Good morning, everyone. So, really, it's my pleasure to welcome and introduce Professor John Tavis. I'm really delighted to have John here today because he's really a good friend for a long time. So we were postdoc fellows at the same time, not in the same labs, but more or less competing in a friendly manner. John graduated at Penn State University, if I remember well, and then moved to San Francisco to the lab of Harold Varmus, who won the Nobel Prize for his work on retrovirology. And he was in Don Gannon's group, and Don Gannon was really a big star of the HBV molecular biology. And John was working on the viral polymerase at that time of HBV, and the mechanism of initiation of reverse transcription. And he was very successful, and then moved to St. Louis University as an assistant professor, and then a professor in the Department of Molecular Microbiology, and really worked a lot. I mean, he had a kind of a bypass with HCV for a couple of years, or maybe a little bit more, and then continued his work on HBV. And he will tell us today about his recent work on the HBV polymerase to find novel drugs to inhibit viral replication, and he's been doing really an outstanding work in that field. So we are really eager to listen to your talk.
All right, thank you, Fabian. So first off, that was embarrassing. Two things I want to say before I get going is, one, I want to apologize for my American's linguistic limitations. I can speak about 20 words of French, and that's not sufficient to give a scientific presentation, so I apologize for making you use my language. The second is, I want to remark at how this is a good example of the best part of the life as a scientist. I'm sitting here looking out, and there's a whole bunch of my friends sitting in this room that I've known for 20 to 30 years. And we've been arguing that entire time, but in any case, they're still very close friends, and it is one of the true blessings of this career. And so with that preamble, I would like to get started. I do have one disclosure. I am an inventor on a number of these compounds that are reported. And if I'm fortunate enough, they may eventually end up as drugs. So let's hope. Okay, I'm not going to introduce HBV here because I figured this audience knows a little bit about it. But I am going to start about HBV replication, which has become kind of a backwater of HBV research recently as things have moved on. So I want to make sure that everybody is on the same page when we get started here. So obviously, reverse transcription copies the viral RNA into DNA, and then it makes the second strand of DNA. So it is a two-phase process. You make a single strand of DNA, you make a double strand of DNA. This is catalyzed by two enzymatic activities on the polymerase protein. These are the ribonuclease H and the reverse transcriptase domains itself in the block structure of the polymerase that's shown there, with the key catalytic residues indicated for all of them. And of course, tyrosine 63 is the priming residue that starts the DNA synthesis. It makes mature partially double-stranded DNA in the cytoplasm of the hepatocyte. So you start out with your RNA, you make single-stranded DNA, you make double-stranded RNA. This all occurs in the cytoplasm. Then that has a choice. It can either be enveloped, go through the intracellular membrane compartments and be released non-cytolytically as a virion, or at least early in infection, you can also have what's been inappropriately called a recycling pathway, leading to the dumping of the DNA back into the nucleus to maintain the cccDNA. It is quite an open question as to how much this operates once a cell is stably infected, but at least early on it is involved in amplification of the cccDNA. But importantly, if you block viral reverse transcription using either nucleoside-nucleotide analogs or ribonuclease H inhibitors, you block it in these stages here, and so you block both of those processes. So it's an attractive drug target. Okay, so our primary target is the ribonuclease H. So why am I concerned about this? Well, this is what happens to the reverse transcription pathway. This is with the nucleic acids drawn out in their linear form. They're actually folded up into like a snarled knot of nucleic acids. But when they're in their linear form, they look like this. If you block the ribonuclease H, what happens is you block the destruction of the viral RNA after it's copied into DNA. So the RNase H and the RT activities work as a concerted enzymatic machine, with the DNA polymerase being about 15 to 18 nucleotides ahead of the RNase H active site. And the RNase H active site acts like a Pac-Man. I may be dating myself. How many of you young folks remember the video game of Pac-Man? Okay, there's a few here. All right, good. Yeah. Okay. All right. In any case, it goes like waka, 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 and it destroys the RNA right behind the DNA polymerase, making a very short overlap between them. If you block the RNase H, that activity doesn't occur. You get these really long stretches of RNA-DNA heteroduplexes, and then you completely block the ability to produce plus polarity DNA because the minus polarity strand is not complete and it's covered up by the RNA. So it's a terminal failure in reverse transcription. And so that's what happens here. And so it's a good target, but no drugs exist against it. And the primary reason for that is working with it is a royal set of difficulties. This is a very difficult enzyme to work with. The first time I did an RNase H activity was in 1992. I did an assay. And yes, I failed miserably. Okay. And yes, I've been largely failing miserably at this the entire time since then. But this has become my quest in my career, and I swear I'm going to get this problem solved before I retire, which means I got to get this done pretty quick, because you can see I'm not exactly a spring chick anymore. So in any case, that's the main target that we're going to work with. Okay, so here we just get out. This is my life, okay, because I have decided that I want to solve this problem. Come heck or high water, I'm going to solve this problem. But after all these years, we have never made a recombinant HBV polymerase derivative with usable RT activity that I can actually measure, do some studies, do some biochemistry on. Complete abject failure. So those of you who are in the lab and you have an experiment that just doesn't work, and your boss goes like this and gets very professorial and looks at you like that, just get used to it. That's your life as a scientist, okay? And the RNase H, we've had moderately more success. We can produce active enzyme sometimes, but they're really a problem. 90% of the preps we use fail in the first place, okay? So the successful preps are not useful for biochemical assays. Their Km values are all over the place, their Vmax values are all over the place, and their specific activity is Lord knows what, okay? And so, yes, we can do it. We know we're close, but we can't quite get there. But I've literally burned out two postdocs and a grad student on this project already. However, I fortunately recruited Dr. Razia Tajwar. She is a stubborn lady, okay? And she has stuck with this project for 5 years so far. And wow, I'm impressed, okay? In any case, the point where we're at is we can only do qualitative assessment of RNase H activity at this time. None of the biochemistry they want to do, and certainly not studying the concerted RT RNase H activity that you really need to do to understand the replication machine that copies this virus. All right, so let's start with some better news now. Google actually did something good. I know this is a shock to everybody in the room, but they have a subsidiary called DeepMind, and they generated a ab initio protein folding program called AlphaFold. I'm sure many of you are aware of it. I love that program. Okay, it makes me very happy because after literally 25, 28 years of trying to produce the protein in a manner that I can get a crystal structure from it, I typed in the sequence and 5 hours later it spit out that structure. And I'm going, that was easier. I like that. Okay. But then of course you look at it and my first thought was, boy, that's a pretty picture. What the heck does it mean? And so then I redirected every single person in my lab onto validating that structure for 3 months. And we used all of the historical data that I was aware of about either the duck hepatitis B virus polymerase or the HBV polymerase and mapped it onto this structure, and every single bit of it made sense. So where we're at is that structure is basically right. But what we don't know is its resolution. Is that resolution consistent across the whole thing? And we also know of at least 5 conformations this enzyme can take just during the initial stages of binding to the RNA and priming. And we don't know which of these structures it is or if it's an average of those. Those are all unknowns. But still, it's a huge step forward and has really helped us understand what's going on here. So the goal of the first portion of the chat that I'm going to tell you about today is to use this model to guide biochemical engineering of the protein to get some enzyme that I can do some proper science with instead of just, getting frustrated. All right, so tell you the first molecule we tried to make is called mini H-Pol. And this is based on a product made by Zhiming Hu, another of our very good friends. He made DHBV, duck hepatitis B virus, mini RT2, and that can specifically prime DNA polymerase. So it's a good assay for studying the priming assay. But of course, nowadays, the way the field has advanced, studying a duck enzyme and saying this applies to the human, it's not getting me a grant at the NIH. So I need to work on the HBV enzyme. So we're trying to use this model to engineer a analog of HBV. So that would, for the priming assay, that would set me up for the enzymatic analyses and would set me up for drug discovery on it. And so we've used our structure-guided protein design to make a mini H-Pol that is based on mini RT2 as closely as possible. And well, that was the first thing we found out was as nice is that it's stable and you can purify it. Okay, right off the bat, that's a success that we don't normally get when we're engineering this. We can purify that every single time, and it always looks like this with about 90 to 95% purity. And it does some activities. First off, this is a fluorescence polarization RNA binding assay comparing binding to the human epsilon RNA and the duck DHBV epsilon RNA. So you can see it's binding the RNA with a reasonable affinity, in the nanomolar range. That's not a bad affinity going on there, but you can also see that there's no specificity for the HBV enzyme. Okay, duck HBV and human HBV are binding with about the same affinity. And we know darn well that human epsilon can't be primed on from the DHBV enzyme and the other way around. So this is a non-specific binding reaction. It is probably reflecting the initial reaction, the initial binding that we know is a non-specific step between epsilon and the polymerase. Then we went into the priming assay, and what this is an SDS page gel that is run out of the protein, and then you're looking for transfer of P32 onto the enzyme by the priming reaction at Y63, and so that makes a covalent adduct. And if your nuke is hot, then the enzyme gets hot. And so that's what this is, and it looks cruddy because this wasn't designed for publication. This was just regular every other day work experiment. But if you look at these last few two lanes there, you can see that yes, it is priming. Okay, that's good. All right, we've got something going on, but we got a huge problem here. One is it doesn't require epsilon, so it's an RNA independent reaction. And secondly, it only acts in the presence of manganese. Okay. That's now what normally happens with DNA modifying enzymes is manganese increases the activity and reduces the specificity. That is a very typical case. So that's exactly what we're seeing here. So what we're seeing is something that we've discovered before, is a cryptic priming reaction. Under certain circumstances, this enzyme can prime at other tyrosines, or we've never seen it prime at anything else, but it can prime at other residues. And this is what you're seeing here. So what this tells me is this enzyme is almost in the right conformation. It can be catalytically active, but it's not doing it right. So, okay, great. Let's do some re-engineering of it. And so I just got a slide deck from Razia this morning. I didn't put it in here, but in any case, she's using molecular dynamics of the predicted structure of this to try and figure out why it's getting trapped in this incorrect conformation. And she's making some good progress in the predictions of for more than just a static look at a single conformation of it. So in any case, I'm pretty pleased with that. So let's shift gears to the RNase H, which will be largely the topic for the rest of the chat here. And so here again is the full structure showing the structure. Now, from the primary sequence, if you look at what has always been considered the RNase H domain based on sequence homology, that's the stuff in green. If you take a look at what the real structure of this is, you can see there's a core of green there, but there's yellow there. That's the thumb subdomain of the RT domain of the polymerase. And this red stuff over here is the N-terminus of the polymerase. That's the N-terminus of the terminal protein domain. It wraps all the way around the enzyme. There's tyrosine 63 right over the DNA polymerase active site. And then it loops around and forms the backside of the RNase H domain. There's absolutely no way we could have predicted that from the linear sequence. And so this model, that's a really good example of the power of the model. The other thing that it shows is if you zoom in on the active site, if you zoom in right about here, one of the catalytic residues, the last of those aspartate acids, aspartic acid 788, is on a flexible loop in the HBV enzyme. You look at the same sequences on HIV, it's on a rigid helix. The rigid helix is what's seen in all of the RNase H's that are out there. And so this holds the enzyme in the right spot to coordinate those two divalent cations, usually magnesium ions there. And so there's a good prediction right there that that's one of the reasons why the HBV is not working properly. Now, in the natural virus, that's got to be held in the right spot, most likely by the chaperone molecules, the HSP90 chaperone class, which is coating this whole enzyme in its native form. That's probably what's holding it in place. But they're not there in the recombinant form. And every time we've tried to complement production of it with human or chicken or whatever species of chaperones, it hasn't helped. And so we have to find a way to lock that into the right spot. And so Razia has systematically changed the residues here that are blocking it from forming a helix, which are these prolines, because prolines cannot be in an alpha helix. It's biochemically impossible. And so she made those changes, and she also looked and noticed that we're not expressing the entire domain. So with those pieces of information, she started mucking about to see if she could fix the enzyme. So the first one that she did was a pair of enzymes. One of them is she just, of those single residue substitutions she put in, proline 790 to L to leucine turned out to be useful. And so that one was put in there. And they also tried designing an RNase H stabilized version where we took pieces of the terminal protein domain and a little bit of the RT domain to try and stabilize it into a true globular enzyme. And when we did that, both of these could be expressed. They could be purified routinely. And if you take a look at the RNase H activity here, this is what we normally see with the wild type enzyme. As pathetic as that is, that's one of our successes. Okay, you can see its early enzymatic activity is not bad. Its initial rate is not bad. It's similar to these other two enzymes, but it dies right away. But you can see if you take the P790L, it has a sustained, robust activity going on there. And then if you take the RNase H stab too, you're still better You're not as good as the P790L in this particular assay, but it has sustained activity too. So we've stabilized the enzymatic activity to the point where we can start to use it. And you can do an inhibition assay with one of our inhibitors, and it makes sense. But we still have a problem, and that's revealed here. If you do a thermofluor, which is a thermal stability assay for an enzyme, because these are maltose binding protein fusions, you can see here, that's the melting point for the maltose binding protein section of this chimeric molecule. So ignore that. Here's the melting point of the polymerase. Here's, if you take a look at the wild type, it's melting at about 17 degrees, which is a chilly lab temperature. Okay, so basically our enzyme is falling apart at room temperature. But if you take a look at RNase H stab, its temperature has moved up, its melting point has moved up to a typical room temperature, about 22, 23. So that's better, but it's still not good because last I heard this enzyme works at 37 degrees. And so we've made progress, but it's not good enough. And so next, Razi asks is, do we need the full RT domain? And so here, I actually designed this one. I'm very proud of it. So in order to make this, given the goofy way that those sequences drape over each other, I had to take the extreme N terminus, the sequences at the extreme N terminus, move them to the extreme C terminus, put a linker in there of a flexible hydrophilic linker of a very precise length in there. Save a little bit of the spacer domain because there's a helix there that's got to be part of the RT domain, and then keep the rest of the TP domain. So I did major surgery on this protein, and when she expressed it and showed me that it works, I nearly fell over. I thought, I mean, you don't do that to a protein and have it work, okay? But it did, and so we were pretty happy about that. Do the thermal stability, and you can see the thermal stability is starting to rise again. But again, you're not getting a clean peak. That tells me that we've got a heterogeneous collection of conformations that are melting at different temperatures. So it's good, it's going up. Some of them are getting up to in the mid-30s, so that's good, but some of them are still down around 20, and it's heterogeneous. All right, so that's a step forward. Then we look at the RNase H activity. So this fraction here, that probably the fraction that's more stable, has got some pretty decent activity there. Again, you can see it's nice and stable. The reactions here are running for almost 2 hours, and it's still maintaining activity. It's a little bit better than P790L. So, okay, good. We're getting someplace. So this currently is our best version of the enzyme, and we're working at trying to improve it a little bit more and to really learn what we can do with it. And also, this has an active RT well, it has an intact RT active site in there. So we simply haven't had the time to explore the RT activity on this, but it's possible that we've gotten where we need to do to have the concerted activity of RT and RNase H activity at the same time. So I'm fairly pleased with this right now. Razi is writing this work up as a paper. She doesn't like writing, so I'm having to be the mean boss and kind of lean on her a little bit. But she's also very good at resisting my instructions. So, okay. All right. So another thing we can do with the model is we can get some structural insights of the RNase inhibition. So as I'm going to tell you starting on the next slide, we have a number of fairly good ribonuclease H inhibitors that we have found over the last few years. And so here you can see a computational docking of one of these compounds into our active site, showing the 2 magnesium sites that are directly involved in the catalysis, showing how the molecule the inhibitor drapes across the enzyme. All right. So what you can see from here is the active site groove, which is right there, is very shallow. And this is a common feature of RNases H. This is just very, very shallow active sites, which makes drug discovery against them difficult. Usually you like a nice deep pocket to tuck a molecule into. But our inhibitors dock into the active site with really quite plausible binding poses. These are measured with estimated binding KDs, and you can see those KDs are pretty decent. These are, of course, predicted ones. And the KDs, the docking scores actually correlate when we plot them against the efficacy against viral replication with fairly decent efficacy. So this is telling us that although we can't, the model is not a high enough resolution right now to really guide atomic level design at the larger scale of, is there a hydrophobic pocket over here? So maybe I should put 2 or 3 methylene linkers in there. It's good enough for that. So it's helpful. It's not perfect, but it's helpful. So now I'm going to talk drug discovery for pretty much the rest of this talk. And so we started with RNase-H screening studies about 12 years ago now, once we got the first version of the enzyme active. And the enzyme was really bad, but it was just barely good enough to start drug discovery. And I found out I really love this stuff. This is fun. It's fun trying to figure out how to make a drug. And so we got into it. And here's a number of different chemotypes. Those are chemical families of molecules that we found. All of these were started, except for this guy, because HIV RNase-H inhibitors are found in these families. Okay. This guy was actually the hit compound for baloxavir, which is the newest influenza virus drug that's out there. And so you can see they all share a trident of chelating motifs here that are involved that can chelate the 2 magnesiums. So the way they work, so I go back here a little bit, you can see it chelates the 2 magnesium ions in the active site and gets in the way so those magnesium ions can't do their portion of the catalysis reaction. So we've screened a whole bunch of molecules here. And so far, what we found out is that this chemotype here, called the N-hydroxypyridine diones, also known as HPDs, those are the best ones we've got. These have got some real potential here to develop. All right, so this just shows a table from my most recent PhD graduate, Molly Woodson's work. Molly was put on this world to be a research pharmacologist. That, the deity has just reached down and tapped her and said that you shall be a research pharmacologist. She is really good. Very nice lady, too. In any case, this is a study that she did showing a bunch of these different HPDs that are sorted by their chemical structure, depending upon what their R group sticking off of it is, showing the efficacy, cytotoxicity, and the ratio of those, and cytotoxicity in primary human hepatocytes. And what you can see is the EC50s range from 61 nanomolar to 7.5 micromolar of this set that you looked at here. And in EC50s, that's effective concentration 50%. That's how much molecule it takes to reduce replication by 50%. Lower numbers are better with EC50s. So that's really good. So just to give you a calibration, in this assay, lamivudine, which is, of course, our approved drug against HBV, has an EC50 of 400 nanomolar. So we're way better than lamivudine already. And then cytotoxicity, CC50, that's how much compound it takes to kill half of the cells in whichever assay you're using. There, as far as drug discovery goes, higher numbers are better. And the ratio between those two is what you want to optimize. And that's called the SI, or selectivity index. You can see that's found in this column right here. And you can see some of those numbers are pretty respectable in that area. And of course, the best one that we've got is there, it's compound 1618. You can see it's a HPD. It's got an EC50 of 61 nanomolar and an SI of 1071. Just to give you a little bit of calibration on that too, Tylenol, or acetaminophen, is one of the most common causes of acute liver failure. It has an SI of 2. Okay, if you take twice the dose of acetaminophen you're supposed to, you're already starting to push on your liver a little bit. So you can see we're a little bit better than a drug that can be sold over the counter. So we're really pleased about this, but this is an inhibitor. These are not yet drugs because we haven't built in the safety and the pharmacological parameters that are needed to convert a good inhibitor like this into a good drug. So that's the next thing that we're trying to do. But there's one other thing I want to point out before we move on, is our standard assay uses HepDES19 cells. These are like the Hep8038s. They are tetracycline repressible, stably transformed HepG2 cells. They're a great tool because all you do is you remove tetracycline from the medium, and every cell in the dish simultaneously starts making HBV. And if you're studying something that inhibits viral replication, it is a superb tool because it isolates that portion of the viral replication cycle. And so the EC50s are the direct EC50s against viral replication. But we all know that viruses are far more complicated than that. They amplify themselves. They do multiple different steps. They get in, they come back out of cells. And so here we tested this in a true infection system. These are HepG2 cells that have the NTCP receptor in them. We did an infection. We added compound right away. And then we waited for a couple of days refreshing the compounds because you don't know how stable your compound is over time. And then harvested it and measured the amount of, measured the EC50s. We did compounds from 3 different chemotypes. You can see here, these are our standard EC50s used with the transfection system. Okay, those are some fairly decent numbers. I mean, these are mediocre here, but they're just representative of molecules. Here's what they are in the infection system. And what happens is you can get up to a 30-fold increase in efficacy by going into the infection system this way than by just doing isolating on the replication assay. And the reason is these are blocking cccDNA formation. They're blocking that initial amplification of the cccDNA and then keeping it down. And so you're getting a double hit here because in the infection system, you have that initial ramp-up phase. We're blocking that ramp-up phase by blocking viral DNA replication. Okay. So in any case, that's really good. So this is an HPD. So if you divide our 61 nanomolar by 30-fold enhancement, if we get around to doing that experiment with that there's a possibility this is a single-digit nanomolar inhibitor. And so that's good news. So what else do we know about this? Because there's a lot of work to turn an inhibitor into a drug. So we know they have moderate to long half-life values in microsomes and human hepatocytes. They have, unfortunately, many of them have stability issues upon storage. This has been our biggest problem that we've been facing is some of the data gets inconsistent. And so we're working to engineer around that, working with our medicinal chemists. These are completely non-toxic in primary human hepatocytes, probably because the primary human hepatocytes are non-replicating cells. And replicating cells are always more sensitive to various forms of toxicity. Most of them have high passive permeability into the cells, so they probably just diffuse into the cells. That's surprising given how hydrophilic they are, but that's what the data says. They do not substantially inhibit some of the CYP450 enzymes. 3A4 or 2C9 are not inhibited. These are important to discover because if you inhibit these guys, these are the main classes of enzymes that detox drugs. And if you inhibit these, you're going to change the half-lives of any other drug that somebody is taking. And so it can cause drug-drug interactions. So 2 of the main ones that are used we don't have to worry about, but it does have a mild inhibition of 2C19, which we have to pay attention to as we move further into drug discovery. Its plasma protein is high. Plasma protein binding is a high that reduces its effective concentration in the blood, but it's okay to work with because obviously we're getting some very good inhibition. They are synergistic with the nucleic acids. You can, with the nucleoside-nucleotide analogs, it can increase the efficacy of these compounds by about 10-fold. And so we're really pleased to hear that. Probably what's happening is you'd think right off the bat, because, hey, I'm hitting DNA replication, that they would be antagonistic because one would get in the way of the other. But they must be blocking the translocation stage or something like that, causing the entire concerted reaction to work less well. And again, this is one of the main reasons why I want to get the concerted reaction working in a biochemical assay, so I can figure that out. Okay. And we have taken one compound. It was the dumbest experiment I ever did, but it was paid for by a different company. So I got a $120,000 free experiment done. So I did it anyway. So we tested the unoptimized HIT compound 208, which is nowhere near as good as these compounds that we've had, in the humanized mouse model, in an infected humanized mouse model. And it reduced HBV replication by 1.4 logs after 2 weeks of treatment in humanized mice. And then when we removed the compound, the virus came back up, which is exactly what you expect from a short treatment with a replication inhibitor.
Monotherapy. So in the mouse was only your compound without nukes.
Without nukes, yes, that was monotherapy in there. And so this is proof of principle that RNase H inhibitors can work in mice. We have to do this experiment properly now. We're gearing up to do it with an optimized molecule where we actually do the PK studies in advance and everything. We figure out the right formulation. None of that was done. We just guessed, but I was shocked that it worked at all. Okay, so that we have 3 compounds that are moving into those pharmacology and efficacy studies. We've tried one PK study, but we got kicked in the shins by the stability issue. The Postal Service decided to take 3 months for the larger batches of these molecules to get from Greece, where they were made, to my lab in St. Louis, and they died during shipment. Okay, and so we had to try it again. And so that slowed us down by well, 3 months. Okay, so that's the summary of where we're at standing. For all of the in vitro type these look really good. Things change when you go in vivo. We shall see. Okay, they're looking good now, but they may not look good in a month from now. We just don't know. Okay. But one of the things that came off of that was a really interesting observation that came from my student Daniel Bradley, who graduated a little over a year ago and is now in Alex Ploss's lab at Princeton. And we just wanted to do the controls to make sure that we knew what we were looking at, that these are actually inhibiting the RNase H activity. So we started just methodically going down and checking various aspects of it. And then we got to the capsid formation. So we did some capsid blots, measuring the accumulation of capsids. The prediction is that this was going to fail, that there's going to be no effect. And well, okay, then we know for sure that that's not a problem. We're just doing our controls, being good boys and girls. But we were really surprised when we did this assay. We being the royal we, of course, I didn't do any of this. Daniel did it. If you take a look here, the assay was we have HepDS19 cells making HBV. We had a compound for 5 days. Then we just made clarified protein lysates. This is just a cytoplasmic lysate. And then we did a Western blot and a capsid blot with the same anti-core antibody, measuring core protein accumulation and capsid accumulation. Very standard assays. I know many people in this room do them on a routine basis. So here you can see is what happened. Here are some one chemotype. These are some HNOs. You can see they reduce capsid accumulation by about 50%. Here are the HPDs that are clustered here. They all reduced, and statistically significantly reduced, the concentration of capsids that accumulated by about in half. And they also had an effect on core protein accumulation, but it was much smaller and much more erratic. And so, but this was a stable phenotype. And so we were looking at it and going, okay, let's chase this one down a little bit and see what's going on. So Daniel spent a lot of work going at it, and I'm taking 2 years worth of work and summarizing it in about 4 slides. So this is what PIs do. So those of you who are junior investigators, just get used to it. You will sweat blood for 1.5 years, and your boss will explain it in a slide, and that's again your life. Okay, and so here what you can see is our primary hypothesis is because we knew these compounds are RNase H inhibitors. So if you block RNase H inhibitor activity, you end up with these long heteroduplexes. Biophysically, heteroduplexes are a lot stiffer than either a single-stranded RNA, a single-stranded DNA, or a double-stranded DNA. They're a lot stiffer. We also know from studies primarily coming out of Adam Zlotnick's lab that the capsid protein is metastable. It kind of flexes and wiggles and moves, and it can pop open really easily. So our hypothesis was the reason we were getting less capsid is because we were building up RNA heteroduplex, getting a tight spring in there and popping it open during the preparation of the capsids, because that is a little bit hard on them. And if you're hard on an extraction, you can pop capsids open. Okay. All right. So we tested that by having 4 different mutations or 3 different mutations that block formation of heteroduplexes by 3 different manners. B702A mutates the RNase H active site, has no RNase H activity. Delta bulge is in the epsilon. This blocks encapsulation of the pregenomic RNA. You get no pregenomic RNA, obviously you can't make a heteroduplex. And YMHA knocks out the DNA polymerase active site. If you knock out the DNA polymerase active site, no heteroduplex. So all three of them do the same thing as far as what we're looking at. And in all three cases, you can see we got a suppression of capsid accumulation. And this is compared to GLS4, which is a CAM-A, or it causes aberrant capsids that fall apart. Okay. Note here also that the activity level here is a little bit lower than what I was showing you before. That's because we found out from additional studies, this expression system that we have produces about 10 times as many capsids as does the stably transfected version. And you're just overwhelming the system by making more capsid protein. So we know that from titration studies. Then Daniel went on and he made just a core protein expression plasmid, just makes core alone. Did the same experiment, bingo, the HPD was suppressing accumulation of it. So this doesn't require any DNA polymerization. It doesn't require any nucleic acids. It's acting on the capsid. We don't know yet if it's acting directly on the capsid or through a cellular intermediate, but it's on the capsid. So that was interesting. So the next thing we did is I called up my friend Adam Zlotnick, and I don't know if any of you know Adam Zlotnick, but he is a really, really good biophysicist. He is the father of the CAM. The entire CAM field was pushed into existence by his understanding and science. And he's really good at this. And he's got biophysical and biochemical assays to study this very well. So the left study here was done by Daniel. And what he did here is he did a titration of the best compound we had, 1466. And as you see, as you get into the low micromolar concentration, you end up with about an 80 to 90% suppression in capsid accumulation. But then in the same preps, he split them in half. And in the other half, he isolated the RNA from the capsids, destroyed all the nucleic acid outside the capsids, then popped them open and did qPCR on the pregenomic RNA that's within it. We stopped production of DNA by including a high amount of lamivudine in this reaction so that we weren't, converting the RNA to DNA and causing a problem. And here you can see it doesn't matter how much compound you put in there, you're having no effect on that. So you're getting rid of 80 to 90% of the capsids, but not affecting the pregenomic RNA in the slightest. Well, in these systems, about 80 to 90% of the capsids that are produced are empty. So what this tells you is it's very efficiently wiping out the empty capsids and not hitting the nucleocapsids that are the ones containing nucleic acid at all. So this is really interesting. Now, whether this has any drug implication, well, I do not know. But in any case, biochemically, this allows us to separate with a reagent that can just be added to a system, capsids from nucleocapsids. So that is at least a good tool already. Then this is an experiment that was done by Caleb Volkner in Adam Zlotnick's lab. What this is a biochemical capsid assembly reaction. So he purifies a whole bunch of capsid, he puts it in a tube, and then he adds salt to 300 millimolar. That causes the capsid to spontaneously assemble in the test tube. And then you can measure the assembly by looking at light scattering as you go from a soluble protein dimer into a big capsid structure. And so if you just add vehicle, that's what you get. If you add our compounds, and there's no dose dependency here, so this reaction got saturated even at the lowest concentration we used in this experiment, you can see that reaction rate is cut in about a half. So this slows the biochemical assembly of capsids, telling you that these compounds are working directly on capsids, because the only thing that are in there is water, salt, capsid, and our inhibitor. That's it. That's all that's in these reactions. The really interesting part about this is that if you add any of the other known CAMs in the entire world, it goes the other direction. It accelerates the capsid assembly rather than suppressing it. So these are the first known molecules that specifically inhibit capsid assembly. So we're going the opposite way. We've got a different effect going on here. Again, I have no idea if this will ever have any impact on drug discovery, but again, it's another good tool for understanding capsid biology. And so then we wanted to compare it to the main classes of capsid inhibitors. Fabian was one of the key authors of an article that named them CAM-As and CAM-Es, which is something that I really want to say thank you for, because I can remember that rather than I can remember class 1 or 2, which I never could remember. So CAM-As, aberrant structures, they make goofy structures that fall apart. CAM-Es make normal looking capsids, but they simply have polymerized so fast they don't have time to get the pregenomic RNA into it. Okay, and so you end up with empty capsids that are stable, but don't have any RNA in it. So both of them block viral replication, but they do it by slightly different manners. But in both cases, they accelerate the rate of capsid assembly. Okay, so GLS4, excuse me, that's a CAM-A. So here, if you take a look at what's happening at capsid accumulation, it's going down in a dose responsive manner. If you take a look at what's happening in encapsulated RNA, those two look a lot alike. So the capsids that can form in that circumstance do have RNA in them, and this is known. That's the normal behavior for a CAM-A. NBR3778 is a classic CAM-E. Here you can see it is having no effect on capsid accumulation, but it's having a dose-dependent effect on the encapsulated RNA. Classic CAM-E behavior. This is 1466. Here's what you've got. Yeah, you suppress capsid, and you have no effect on RNA, just like I showed you on a preceding slide. So what we named these is CAM-Is for CAM inhibitors. I don't know if that name's going to stick. Hope it does. But in any case, this is the tentative name for these now, and this is the one we're at least using in the lab. So it's a new class of CAMs that we found. All right. So what we've done since then is, because Daniel had this really obnoxious habit of graduating and leaving my lab. Okay. I mean, talk about inconsiderate. Okay. He knew all these assays. He was working fast, and then he left. Oh, man. So in any case, we've been slowed down on this. But my very talented lab manager, Dr. Shalon Lee, has been following up. And so she's now screened another 93 capsids in the capsid blot assay. As you can imagine, that's not a high throughput screening assay. You're not going to do 10,000 of those, okay? But she screened 93 of them at least once. 29 are confirmed. And so I'm showing you the data for the confirmed molecules here. So in the HPD class, these are the ones we've been concentrating on because we know we've got interesting molecules in there. She's found 11 additional ones that suppress capsid accumulation. So this is, it's 11 out of 18. So roughly half-ish of those molecules have this additional activity. And none of these suppress capsid without hitting the RNase H. The dual arrow means inactive against the RNase H activity. Okay, so none of them fall into that case. We haven't been able to separate those two activities for the HPDs yet. There's another chemotype that doesn't look anything at all like the HPDs. We've screened 6 of those. We don't have many in our library, but half of them suppress capsid, and none of those are active against the RNase H. We don't know if those are just a regular CAM or if those are CAM-Is yet. But in any case, we have other molecules that can also affect the capsid in our library. And then a couple other chemotypes we're poking at, and none of those have hit yet. And so the next step is to figure out if these new molecules, or actually even these guys, how many of them are CAM-Is and how many of them are of the other classes. And Shalon has got about 1 to 2 more weeks of confirming preliminary hits, and then she moves on into doing the pregenomic RNA analyses, which will tell us what we need to look at. So ask me in a month, I'll be able to tell you a little bit more about it. Okay, so that's the chat I wanted to tell you. There's 2 basic stories here. One is that predicting the HBV polymerase structure has enabled protein engineering. It is improving recombinant polymerase activity. So, but it's not. We're still not where we wanted to. And so, literally, I've been working at this since 1992, and I'm almost there. So, maybe by the time I retire in, 3 to 5 years, maybe I'll get there. And so we'll see. We have RNase H inhibitors that are really pretty good in vitro. Again, these are inhibitors, not drugs. They're very active in infected cells and it can suppress formation of cccDNA. We explicitly demonstrated that. The in vitro pharmacology studies are showing no red flags. They look okay for this stage of development, but we're a long, long way away from starting to think about doing a phase 1 clinical trial. Okay. Some RNase H inhibitors can inhibit capsid accumulation, okay, by a novel mechanism. Again, we don't know if this is just interesting capsid biology or if this is going to have drug discovery implications. If we can get molecules that have RNase H inhibition and an anti-capsid activity, you've got a dual activity molecule and you've got combination therapy going on with a single molecule. That'll get really attractive. Okay, so that's one thing that we're really quite excited to hopefully find. And finally, we've shown in data that I didn't have time to show you, but it's important for the drug discovery concepts, is that they're synergistic with nucleoside analogs and can inhibit HBV replication in humanized mice, at least in pilot studies. So, okay. And so finally, I want to acknowledge the people in my lab whose data I talked about today, the ones that are grayed out, have moved on from the lab and are all doing very well in the next stages of their careers. I'm very proud of them. Here are these are the 4 current members of my lab, current full-time members. There's a flock of undergraduates that come and go, and you never can tell what they're doing. But in any case, they're around. But these are the 4 full-time people in my lab. And then Marv Myers is one of the medicinal chemists that I work very closely with. And the other medicinal chemist I work very closely with is Grigoris Zoidis. And he is at the University of Athens. This has been funded by a number of grants over the years. And those are just the numbers of them. And so with that, that's what we've been up to for the last couple of whiles, like since 1992. And I'm happy to take any questions you may have.
Really outstanding discussion. Thanks a lot, John.